Generally, enzymes are mostly utilized in the paper and pulp business, textile and pharmaceutical industry, as well as the food sector. Enzymes are proteins that catalyze biological reactions and can be found in all living creatures, which usually be used in food processing like making the beer and wine, purifications and filtration, baking, cheese making, and much more. Food and beverage manufacturing is a significant market for industrial enzymes, particularly for enzymes used in the food and beverages manufacturing. Food enzymes are increasingly used in industrial food processing, which usually obtained by microbial fermentation, which are wild-type and genetically modified microorganism strains. By optimizing the fermentation process, the production of food enzyme can be increased by utilizing GMM or by generating recombinant enzymes. Therefore, the industrial production of food enzyme preparation and a particular on the utilization of GMM will be discussed in this video.
So today, I will explain to you about the industrial production of food enzyme. There are 4 steps to produce food enzyme in industry. First is fermentation, second is purification, third immobilization, and fourth formulation. Firstly, fermentation process is a process where the microorganism convert an energy source into other substances. Fermentation is the only first step in production of a refined food substances using a microorganism regardless of whether that microorganism is genetically engineered. Food enzyme can be obtained by microorganism through fermentation or from animal and plant by extraction. There are different types of fermentation process can be used such as submerged fermentation and solid state fermentation. Therefore, different types of submerged fermentation exist: batch, fed batch, perfusion, or continuous culture method. During solid state fermentation, microorganisms grow on solid substrate and are often preferred for fungal fermentation. Secondly is purification. Purification is one of the downstream process to obtain purified enzyme concentrate. Different purification steps depend on the type of fermentation used. For enzyme produced intracellularly, cell wall and membrane need to be destroyed first by mechanical or enzymatic method, while for extracellularly, the first purification step is consists of dispersion of cell from fermentation growth. Third step is immobilization. Soluble enzyme and immobilized enzyme can be used during food production. Traditionally, soluble enzyme have been used and the product need to separate from unused substrate and the enzyme at the end of the process. The separation often causes a denaturation of the enzyme, so they require the use of new enzyme. Therefore, immobilized enzyme allow the reuse of enzyme for multiple batch with low cost and very suitable for industrial scale production. And the last step is formulation. There are 3 different forms of enzyme. First, enzyme protein itself, which is pure substance. Second, enzyme concentrate, which is the product after fermentation or extraction. Lastly, enzyme preparation, which the formulation contain one or multiple enzyme concentrate with the addition of stabilizer, preservative, and diluent to stabilize the enzyme. Those 3 forms of enzyme are the formulation that are sold in the market.
Next is the factor that make microbial sources are more preferred in production of food enzyme than animal or plant sources. In general, microbial sources are more favored in the production of food enzyme than extraction from animal or plant sources, which mostly for economic and technical factors. And these are the other factors that make microbial sources are more preferred than animal or plant sources. First, many microorganisms produce enzyme extracellularly, which simplifies the purification process. And even if the microorganism produced intracellularly, the process in obtaining the enzymes are much more easier than using animal or plant sources. Secondly, smaller production facilities are required, which resulting in lower production costs. Animal and plant need to be transferred to extraction facilities, which are not required for microbial fermentation, where the entire process can be conducted in one place. Microbial enzymes frequently had higher activity and more stable, As example, enzymes that derive from thermophilic microorganisms will have higher temperature resistance. Next are the parameters that need to be considered in producing recombinant strain. First, selecting the host organism. Second, the expression vector and transformation method. And lastly, the selection marker used. As for selecting the host organism, the host should contain the required characteristic for the production and expression of the target enzyme. As for bacteria, they are simple and low-cost hosts. They also have numerous expression systems that are accessible for recombination. However, only simple protein can be synthesized due to some issue regarding protein folding. The most often used bacterial strain for the production of food enzyme are E. coli and the Bacillus. E. coli, it consists molecular tools that help in production of expression system for higher yield enzyme production. And the Bacillus, they produce higher enzyme concentration extracellularly. As for fungal host strain, they have subcellular capabilities for proper protein folding and post-translation modification. However, they only produce a little amount of their own secondary protein into the medium. Therefore, they need further purification step. Most commonly used fungi are Aspergillus niger and Aspergillus oryzae.
Next are the types of expression vectors and transformation method. And expression vectors typically include origin of replication, multiple cloning site in which gene expression will be integrated, and selection marker for selection of the recombinant strain. There are three types of expression vectors that being used in the industry. Firstly, the integrative plasmid. It is able to insert and incorporate the plasmid into the host genome. Also, it has two integration sites that will make the vector to be cleaved. Second is aposomal plasmid, where there can be extrachromosomal by using autonomous replication plasmid. And also, it requires the presence of origin of replication that is homologous with the host strains. And lastly is shuttle vectors, in which they can be introduced into prokaryotes and eukaryotes. So, to transfer those expression vectors into the host organism, we can undergo any transformation method, such as conjugation, electroporation, the use of computer cells, and vector incubation using protoplast.
Lastly, the selection marker used to identify the modified strain. For bacteria, the selection marker mainly based on the integration of antimicrobial resistant genes. However, due to health issues regarding antimicrobial resistant genes, and autotrophic mutants can be employed. As for fungi, the selection marker used are the autotrophic markers, such as the AMDS marker, where it encodes for the acetamidase enzyme, which will hydrolyze acetamide into acetate and ammonia. And by using acetamide as the sole nitrogen supply, the strain that have been modified with AMDS marker can be screened. Next is the possible strategies that can be used in order to increase the gene expression. First, during transformation, use a large number of DNA. Other than that, by applying a high antibiotic pressure on the selection marker. This can be done by utilizing a high concentration of antibiotic or a weak promoter.
Next, I will present about food enzymes and related GMO regulations. There is issues related regarding the regulation number 1829/2003, which applies if material from genetic modified source is present in food product or enzyme preparation. This poses a problem in comparison with detection threshold of 10 nanogram of DNA per gram or mil stated by European Food Safety Agency (EFSA). As 10 nanogram of DNA correspond to an estimation of at least 10⁻⁶ estimated genome copy numbers per gram. However, the requested performance of the official real-time PCR method used for GMO control in food and feed product has a limit of detection which is lower than 25 genome copies. Belgian enforcement laboratories, with the support of the relevant Belgian authorities, have recently developed a strategy to target the unexpected presence of GMM strain. This diagram shows general overview of the developed strategy targeting unexpected presence of food enzyme producing microorganisms. First, by targeting 16S rRNA sequence and the internal transcript spacer region. The identification obtained by sequencing followed by blasting against in-house database. Next, the presence of antimicrobial resistant AMR genes is targeted using qPCR amplification. As additional analysis, a viability assessment is performed. Using this strategy, a detected modified organism can be identified. This is an example of genetically modified microorganism producing protease. As the application of developed strategy, a few observations were made. First, the presence of bacterial DNA was detected in the food enzyme preparation, which was identified as bacillus. Additionally, the screening of the product for key AMR genes demonstrated the presence of the full-length AADD gene conferring KANR. Valuable bacillus cells were also isolated from this food enzyme preparation. The isolated cells were consequently analyzed again using PCR and qPCR methods to verify the presence of full-length AMR gene.
Next is Nakli and Akli of GMM in food enzyme production. The idea of using GMM in food enzyme production is a knowledge that given by Allah to its human, as in Quran Surah Al-Alaq verses 5. أعوذ بالله من الشيطان الرجيم علم الإنسان ما لم يعلم, which is meant that taught man that which he knew not. And also the usefulness of GMM for food enzyme production, as it can increase the enzyme yield and also the safety advantages. It is one of the findings that was in Allah's blessing, based in Quran Surah Al-Fussilat verses 53. أعوذ بالله من الشيطان الرجيم سنريهم آياتنا في الآفاق وفي أنفسهم حتى يتبين لهم أنه الحق. صدق الله العظيم. This means that we show them our signs in every region of the earth and in themselves until it becomes clear to them that this is the truth. This ayah proves that Allah is the only creator and the only one who can control his creation on earth.
In conclusion, the characteristic of GMM used during the food enzyme fermentation process has been outlined, which include a description of the selection criteria to choose an appropriate host microorganism vectors and selection markers. Moreover, the microbial sources are more preferred to undergo the GMM compared to plant and animals. This is because it's easy to extract from the cell, it can reduce the time and also can reduce the cost productions. Thus, it will increase the production of gene expression to produce food enzyme that will provide a benefit to the industry.